I'm really excited to meet you. My name is Jessica. I'm currently a high school student, but I would like to go into forensics in a job area similar to what you're doing as a forensic DNA analyst. And I've come to your office today to see what this job would be like.
Hi Jessica, it's great to have you here. I'd be happy to show you many of the techniques we use in our day-to-day investigations. The first thing you should know is that in this stream of forensics, we analyze DNA from the crime scenes as well as the suspects to determine if we can find a match. We use a method called DNA profiling to distinguish one individual from another.
How can DNA profiling help to distinguish one individual from another if 99% of human DNA is similar?
That's a great question. Well, in forensics, we use the 1% of DNA that differs between individuals, and these are called polymorphic sequences.
Wouldn't that be an invasion of privacy if those DNA regions code for specific traits?
Yes, it would. And that is why we use regions of the DNA that doesn't code for RNA or proteins and are therefore anonymous. This ensures the privacy of the individual under investigation. So there are 3 types of DNA profiling. The first is called restriction fragment length polymorphism analysis, so RFLP. And this technique is based on the fact that everyone differs slightly with respect to the location of certain restriction sites.
What are restriction sites and how do they help you distinguish between individuals?
So restriction sites are where the restriction enzymes cut the DNA. And once the DNA is cut, the resulting DNA fragments are separated on a gel. Once we stain the fragments, we can compare the restriction fragment patterns between individuals.
Are there any issues with this technique?
Indeed. The main problem with this technique is that it requires a large amount of DNA, and often we don't have a large amount of DNA from the crime scene. So more frequently, we use the other techniques, VNTR and STR analysis.
What allows VNTR and STR analysis to be used when the quantity of DNA is minimal, whereas RFLP analysis requires large amounts of DNA?
Because VNTR and STR analysis use smaller sections of the genome, we can use the polymerase chain reaction, also known as PCR, to amplify these regions of the genome.
I'm sorry, I don't quite understand PCR. Can you explain what it is and how it works?
Yes, for sure. The polymerase chain reaction, or PCR, amplifies small regions of DNA. The procedure was invented by Carey Mullis in 1983. PCR is like an in vitro DNA synthesis reaction that copies DNA sequences over and over, making millions of copies. Even the components we use in the reaction is similar. We use DNA template isolated from a sample, primers flanking the region of interest, heat-stable polymerases like Taq polymerases, DNTPs, and buffer containing magnesium chloride. There's 3 main steps of PCR. The denaturation step is where the DNA strands are melted into single strands by heating it to about 95 degrees Celsius. There's the annealing step, which involves cooling the reaction to about 45 to 65 degrees Celsius, which allows the primers to bind or hybridize to the template strand. And lastly, there's the extension step. The temperature of the reaction is raised to 75 degrees Celsius to allow the DNA polymerase to extend the ends of the primers and form new strands. This process is repeated about 25 to 35 times in a thermocycler.
What portions of the DNA are amplified?
We can amplify anonymous sequences of DNA that differs between individuals. Some anonymous regions have short DNA segments that are repeated over and over, and these are called tandem repeats. The number of tandem repeats often varies between individuals, and it is inherited in a Mendelian fashion.
Can you explain Mendelian inheritance for me?
Yes. Humans are diploid, meaning they have two sets of chromosomes, one coming from dad and one coming from mom. Therefore, an individual can either be homozygous, with the same number of repeats on each sister chromatid, or heterozygous, with a different number of repeats at a specific location or locus on the chromosome.
Okay, that makes sense. Now, what is VNTR and STR analysis?
So variable number tandem repeat analysis, or VNTR analysis, looks at DNA segments ranging from 10 to 100 base pairs in length. On the other hand, STR analysis, which stands for short tandem repeat analysis, is much like VNTR, except the repeats are only 2 to 9 base pairs.
When would you prefer to use STR over VNTR?
We prefer to use STR analysis over VNTR analysis when the DNA sample is degraded or aged. To perform PCR, we need at least a few hundred base pairs around the VNTR region. And because the STR repeats are smaller, there's a greater chance an STR repeat that's intact will end up in the DNA sample.
What do you do with the PCR products?
Well, we take the PCR products from RFLP, VNTR, and STR analysis, and we run them on a gel.
How do gels work?
Gel electrophoresis separates molecules based on size and charge. Because the restriction fragments and PCR products are relatively small, we use an agarose gel rather than a polyacrylamide gel. The agarose acts as a matrix through which the DNA fragments migrate. The larger fragments travel slower through the gel, while the smaller fragments travel more quickly. There are two electrodes, a negative electrode at the side where we put the DNA sample in, and a positive electrode at the opposite end. Because DNA is negative, it is pushed from the negative electrode towards the positive electrode. Let me show you how to set up a gel. So first you need to insert the gel comb at one end of the gel box. Then you mix the agarose with Tay buffer, warm it up to dissolve the agarose, and add a dye like CyberSafe to the molten agarose.
What is the purpose of adding CyberSafe?
CyberSafe intercalates between DNA bases, allowing us to see the DNA bands when we place the gel under UV light. We will see the dye fluoresce green. And once we add the CyberSafe to the molten mixture, we pour it into the gel tray and allow it to solidify. Once it has solidified, the comb is then removed and we set the gel tray inside the gel box with the electrodes at either end. We fill the box with a running buffer or Tay buffer, and we take our DNA ladder and load it into the first well. Then each digested DNA sample is loaded into the following wells, and we run the gel.
How does the DNA sample not disperse in the Tay buffer?
So we add the loading dye to the sample to make it so heavy that it drops into the well. And the dye also stains the DNA, allowing us to see how far the bands travel down the gel. And when they travel about 2/3 of the way, we know to stop electrophoresis, remove the gel from the tray, view it under UV light, and take a photo.
Why do you include a DNA ladder?
We include the DNA fragments of known size into one well so we can determine the size of the DNA fragments of the samples.
Is there anything else you would like to teach me about forensics before I go back to school?
Oh yes, there is one more thing. When we collect DNA information from suspects of criminal offenses, we enter it into a software program that generates a database called the Combined DNA Index System, or CODIS for short. Although its original purpose was to collect information about convicted criminals, Canada and the US enacted legislation that allows the collection of DNA evidence from suspects before conviction. And this system uses 13 loci for profiling. Obviously, the more loci we use, the smaller the chance of an exact match occurring by chance.
Can it differentiate between males and females?
Yes, we analyze what's called the amelogenin locus, which serves as a marker for gender because it's located on the human sex chromosomes. Amplifying this locus produces both X and Y chromosome products of different sizes, and this allows us to determine whether the offender was a male or female.
What are the chances that two people would have the same DNA fingerprint?
To know the likelihood that two individuals share the same DNA profile by chance, we can calculate what is called the random match probability, or RPM. This calculation determines the frequency of a specific genotype in a population. It takes into consideration the individual's ethnic group. And once we have the population frequency of the STR alleles we are analyzing, we can use the Hardy-Weinberg equation to calculate the RPM for each STR genotype. Once we have these, we can multiply them together to get the overall profile frequency. The FBI determined that the probability of two unrelated Caucasians having the same STR profile for the 13 loci is about 1 in 575 trillion. And this means that only 8 or 9 pairs of people in the world would be expected to match at those 13 STR loci.
That is super interesting. This is definitely a field I would like to go into. Thank you, Jane, for letting me come into your office today and sharing your knowledge of the many techniques used in forensics.
My pleasure. Thank you for coming, and I wish you all the best in your future studies and career.
Hello? Officer? A murder at UBCO? I'll be there in an instant. Ciao.
Hello, officer? I think we have a match. Suspect 3.
Good morning, everyone. Please be seated. Will the clerk please read the charge to the accused?
Mr. Smith, please rise. You are charged honor about the 5th of April 2021 at or near UBCO. You did unlawfully kill Matilda Wilson, contrary to the criminal code. How do you plead? Guilty or not guilty?
Not guilty. I did not do it.
Good morning, my lady. My name is I Do Good, and I'm the prosecutor of this particular case, and I'd like to call my first witness, Constable Susan Black.
Very well.
Now, Ms. Black, I understand that you're a member of the Royal Canadian Mounted Police based at the UBCO detachment. Did you call in your crime scene investigators to take photographs and secure samples of the blood and various exhibits?
Yes, I did.
So the next witness, my lady, for the prosecution is crime scene investigator Ms. Jane White.
Your Honor, I am the one who collected the blood off the floor leading out of the laboratory and took skin cell samples from the handle of the knife. My team of investigators found footage of 4 suspects coming out of the lab around the same time that the murder is thought to have occurred. I obtained blood samples from each suspect and a cheek sample from myself. I extracted DNA and did the appropriate analysis for each sample. First, I performed RFLP analysis on the blood sample from the floor of the crime scene leading out of the laboratory. The gel clearly showed that suspect 3 lines up exactly with the crime scene blood sample, CS1. I measured the distance traveled by each band in the DNA ladder, generated a graph of the band size in base pairs versus distance in millimeters. I set the band size on the vertical axis to log scale and used the exponential equation from the graph to calculate all other fragment lengths in the gel. In Table 1, you will find that the measured values as well as the length of the DNA fragments for CS1 is closest to suspect 3. STR analysis at the TH01 locus was also analyzed. For this test, DNA was extracted from the skin cells of the knife handle found at the crime scene, as well as from the blood samples of each suspect. The TH1 genotypes were determined for suspect 1 to 4, as well as for the crime scene. It appears that the TH1 genotype from the crime scene matches exactly with that of suspect 3. To make sure my own DNA did not contaminate the sample, I extracted DNA from my own cheek skin cells and performed VNTR analysis on the D1S80 locus, which you can see in Figure 4A. The skin cells from the knife handle found at the crime scene was also analyzed at this locus, Figure 4B. The same method that was used to calculate the fragment size in RFLP analysis was used here. Figure 5 shows the log of DNA fragments in base pairs versus the distance migrated on the gel. The equation from this graph was used to calculate the size of DNA fragments from the skin cells at the crime scene, sample CS2. In Table 3, you can see that the offender is heterozygous at the D1S80 locus. The same analysis was done on my own cheek cells, and it revealed that I am homozygous at the D1S80 locus. When the size of the bands on the gels in Figure 4 were compared to the allele sizes closest to the estimated band size, the number of repeats corresponding to the allele size was determined. Refer to Table A1 in the appendix. The D1S80 genotype for the crime scene and investigator DNA can be observed in Table 4. The genotype for CS2 is 3355, and my own genotype is 2727. Therefore, my DNA did not contaminate the sample. These results help us to eliminate the possibility of suspects 1, 2, and 4 committing the murder because their profile did not match that of the crime scene. However, DNA fingerprint from suspect 3 did match the crime scene DNA profile. Therefore, it is likely that suspect 3 committed the crime.
Well, lady, I'm calling as a defense witness the accused, Mr. Smith. Now, Mr. Smith, did you kill Matilda Wilson?
Of course not.
You and Ms. Wilson were competing for COVID-19 vaccines. Is that true?
Yep.
What was your evaluation of her work?
Yeah, far inferior.
What hope did you hold out for your vaccine?
Oh, absolute acceptance.
And it's my understanding that she said something to you about her work being superior. Is that correct?
Yes.
At any time, did you have a minor altercation with Ms. Wilson over the validity of her work as opposed to your work?
Yes.
Thank you, my lady. Those are all the questions I wish to ask of Mr. Smith. On behalf of the prosecution, this man ought to be given a substantial sentence in jail. I suggest that he be sentenced to the local penitentiary for at least 25 years, and I request that you assign him to the laboratory to clean out the flasks and sweep up the floors. Thank you, my lady.
Mr. Smith, I agree with the prosecutor. I sentence you to 25 years washing bottles in the UBC laboratory.